Hi guys, welcome to class. Oh. All right, so today we will be continuing with our RNA-seq analysis. I'm going to do a short set of slides on some statistics for differential gene expression and how that works and how we're going to approach that and other ways that you might want to approach it depending on the organism of interest. If you're doing this for your research, I have tons of papers I can give you and lots of things that you can do to explore a little bit more into depth on that. Then Narula is going to pick up with the lab, and we are going to get back on the ACF and try to get everything working. Did anyone try the script I posted? Did it work? Yeah? Oh, good. Okay, great. So if things are not going well with us all getting interactive jobs, we can start trying to write Q subscripts and launch the jobs. When I launched that job over the weekend with a full like 16 threads, it ran in like 4 minutes. It was great. So we could start trying to do that. But hopefully we'll be able to get an interactive session. I finally got around to posting a homework. I got a little behind on things, but so I put this homework up. It's kind of a big homework because we're running a little late. So instead of giving you a week, I gave you 9 days. So it won't be due until next Wednesday. There are some regular expression questions. I would encourage you to go ahead and do that. We're not going to talk about regular expressions again, and that material is going to get stale fast. So go ahead and do that and try, and don't wait till the end because you'll be unhappy. And then there's an RNA-seq question where you have to go get some data, map it to the Arabidopsis genome with STAR, and then give me some statistics and do a little bit of reading of the manual of STAR. And I think these types of questions are really good, even though they're highly reflective of what we've done in the lab, because you need to start developing the self-confidence and the skills to go out, get your own data, do the mapping, go all the way to the end of the analysis by yourself without having a lab to guide you. So this is kind of an in-between where you can look back at what we did last Wednesday and just redo the same thing with a new file and start trying to think about your mapping results. So it's kind of a big homework. I just want to give you fair warning on that so you start early. There are 2 extra credit points, so I'll score it out of 10, but you know, do your best anyway. We have lots of grading coming up, stuff that is graded. So Monday, project proposal due. Just email me a 1-page project proposal. I'm not looking for anything too fancy, but give me an idea of where you're going to get your data, what you want to do to it, what kind of tools you foresee using, maybe, you know, just the general outline of the experimental goals. And then that homework that I just talked about is due on Wednesday, and then the next Monday is our final test. I will get you a study guide by a week ahead of the test. So next Monday, there will be a study guide posted so you can see what I'm going to be looking for on this test. Any questions about all of that? You feel pretty good about that? All right. And I'll be working on grading the current homeworks this week. Okay, so I'm going to talk about differential expression statistics. I get a little bit more in depth on this. Over the summer, I generally teach a 1-week RNA-seq workshop, kind of an intensive. We used to do it 2 days, 9 to 5. Last year, we tried out a 9 a.m. to noon for a whole week, and I think that format was a little bit better. So if you want to come to that, we'll probably offer it next summer, and we get a little more in depth on this. Also, consult a statistician. It's a good idea before you start sequencing. I said that last time, but I want to say it again. So let's talk about how to go from counts to differential expression statistics. So this is what we are hoping to come out of probably by the end of today. We probably won't actually get to the statistics today, But your goal in RNA sequencing, if you want to do differential gene expression, is to take your RNA-seq data and figure out how many of each of my reads aligns to each of my genes. How many can I uniquely assign to a gene? So you're trying to take something that's a lot of raw nucleotides and turn it into something quantitative that you can do statistics on. So for example, and this is the typical format that you're going to put into a lot of stats programs. For gene 1, in my 24 GACL sample, I had 8 reads. In my 24 GACP sample, I had 3 reads. And some genes are more highly expressed across the board. There are usually lots of zeros, so it can be quite a sparse matrix. But this is the goal, is to go from those raw reads to counts per gene. So the first thing we have to do with this data is talk about normalizing. The biggest concern that you absolutely have to normalize for is that you're going to have different numbers of reads per sequencing run. So some of your libraries are going to come out with millions of reads, maybe a whole order of magnitude more than another library. That means you can't compare your read counts directly, right? Because your sample size is different. There are other concerns that are of varying importance. Some people worry about them. Different lengths of transcripts. So if your genes, or even better, if you have alternatively spliced transcripts, are very different lengths, when you do sequencing and sample from them, you're going to sample from a transcript that is shorter less often, because you're going to randomly fragment, you're going to select those fragments. So the length of the transcript is going to matter. Take a single transcript, you can get multiple individual reads out of it, you'll sample it more. You're going to have differing ability to map reads. So some genes have gene family members of big families, and the reads will map to multiple locations. So there may be whole regions of the gene that you can't utilize for uniquely mapping. It's a wash, you have to throw those away. So that differing ability to map reads back to different genes can be influential. GC bias. Your instrument is not always going to sequence through regions of genes that are particularly high or low in GC content. So again, all of these are concerns that you may want to normalize for, or people might want to tell you you want to normalize for. However, I'm going to tell you what my approach, which is you can safely ignore all of these, because what you want to do statistically is compare the gene expression within the same gene across samples. So this just means, for example, different lengths of transcripts. You're going to make the assumption that the length of the transcript in sample A is exactly the same as the length of the transcript in sample B, and therefore any sort of discrimination between those genes is not going to matter, because all you care about is that one gene across samples. I don't know if I'm saying that very clearly. You can wash this out because you're not trying to compare the genes to each other. All of these are problems when you try to compare the genes to each other. If you don't compare the genes to each other, you compare the genes to themselves across samples, you can ignore all of that. You cannot ignore the first one. You have to normalize for the number of reads per sequence run. You cannot get around that. But these other concerns, you can kind of ignore them. And some statistical packages will attempt to handle them for you anyway. But they're not going to matter, because your ability to sequence the MLO gene in your first sample and your ability to sequence the MLO gene in the second sample is going to look the same. And all you're asking is, did that MLO gene change in expression between sample 1 and sample 2? So you aren't asking, is the MLO gene expressed at the same level as kinase 17? That would cause problems, because maybe the MLO gene is way longer than kinase 17. That would cause a problem. But you're not ever going to ask that question. You're only going to ask for each individual gene. It's going to have its own statistical test, its own p-value, and that's going to allow you to ignore a lot of problems, which is nice. Okay, so old normalization methods, you'll probably still see these in papers. Lots of people still love them. I think there are better ways, but people still use them. RPKM, reads per kilobase of exon per million mapped reads, is what that stands for. So let's break that down. You take your read count, you multiply it times 1,000,000, and then you divide that by the transcript length times the total reads sequenced. So this is attempting to normalize for both the length of the transcript and the total number of reads sequenced in this library. This would be a per library metric. So for each individually sequenced library, for each gene, you would calculate an RPKM value using this formula. So very straightforward standard normalization technique. If you have paired ends, you handle the paired ends as a single unit. So you do not handle them as two independent samples because they're not independent. You started with a single RNA fragment, turned into cDNA, and a sequence from both ends. So you are going to treat that as a single fragment, even though you have two reads. And that's where you get into fragments per kilobase of exon per million mapped reads. So the same thing, except you account both of your paired end reads as one thing. The original strategies that people use for normalization have suffered from some significant problems. Read densities, if you only compensate for total read depth, that's not really sufficient to compensate. And so a lot of the thinking about normalization now is, one thing is that a small number of genes can consume a significant fraction of sequencing resources. So if their expression changes, this kind of skews the distribution for everything else. And the way to think about this might be, like you have a leaf, or you have some plants, and you keep some in a control chamber and some you're exposing to a lot of heat stress. They hugely ramp up all these reactive oxygen scavenging genes. So these some genes that were already maybe like moderately expressed are now hugely expressed, and they eat up 50% of your sequencing lane. The other genes in the sample may not have actually changed in expression. They just look like they've changed in expression because now all of your read mapping is for these really highly expressed genes. So what you have to do, what a number of statistical programs are doing, is looking at, instead of normalizing across the total read depth, try to do this a little bit more subtly. So basically, put another way, the problem is the proportional representation of each gene is dependent on the expression levels of all other genes, because you're sampling from, you have a set number of reads that you have to sample. It's more complicated to kind of massage the numbers and deal with these sorts of things. So you usually want to go find a software package to make your life easier, unless you're in a lab that does statistics, genomic statistics for your research, in which case you may want to develop your own package. But if you're not, like me, you want to go find a package that handles this gracefully for you. But basically, you're looking for a package that's going to have scaling factors that exclude or reduce the impact of highly expressed and highly differentially expressed genes. So by getting rid of those and taking those out of the normalization equation, everything else gets normalized in a much more rational way. I have some brief descriptions here. The two packages that my lab usually uses are DESeq and EdgeR. DESeq starts by computing the median of the ratio for each gene of its read count over its geometric mean across all samples. So basically, the important part here is that you can assume that most genes are not differentially expressed. That is actually a dangerous assumption in some situations, because sometimes you're doing something so drastic that the majority of genes are differentially expressed. But if you assume that most genes are not differentially expressed, then you can kind of obtain this rational scaling factor to look at how to normalize genes correctly. EdgeR used, I think they were the first ones to pilot the TMM gene expression metric, which is trimmed means of M values. And again, it's basically excluding genes that have overall very high expression or very different expression, then compute a scaling factor. So they're both using somewhat different underlying statistical approaches, but the goal is the same, which is to wash out the influence of those really either genes that are super highly expressed or that are changing a lot in your experiment. And I think I talked about this a little bit last time. A lot of people use these specifically if they don't have a lot of biological replicates. And this turns out, this is part of the reason why you have to make some unfortunate assumptions that you can measure biological variability that based on most genes not being differentially expressed. A lot of that is because we just aren't generating enough biological replicates. So these are good for if you only have 3 or 4 or 5 biological replicates, but if you can get up to 10 or 12 or something else, you want to look at different statistical packages. Originally, pre-2010, Poisson distributions were generally used to model counts. However, the data was found to be over dispersed. So we see so much more variation than can be put into a Poisson distribution. So that leads to a lot of high false positives. So now most packages are going to use a negative binomial distribution. It's a good substitution for an over dispersed Poisson, kind of a common, if you're a statistician, apparently that makes a lot of sense. But because so the Poisson requires your mean and your variance to be, they're like pinned to each other. But a negative binomial does not. You can have a very different mean and variance. And this is just an example of one of the first papers to propose this, where the purple was the variation implied by Poisson, and then the orange is what they were actually seeing, which deviates significantly at high levels of expression. However, and that's the approach we'll see in this class with DE-seq2. However, there are a lot of other approaches. There's the cufflinks package is beloved by, for some reason, the animal science people always like to use cufflinks, and the plant people always use EdgeR and DE-seq. Why? I don't know. Different communities land on different things that they really like. But cufflinks uses a beta negative binomial, I believe. There are a few packages that still use a Poisson distribution. There's a Bayesian approach that people are getting very excited about. And the non-parametric approaches, I would not use those unless you have a lot of biological replicates. So the SAM-seq and the NOI-seq, or noise-seq, I guess it might be noise-seq. Those I definitely wouldn't use without with lots of replicates. Each one has further variation normalization procedures and testing strategies. So it's kind of the wild, wild west with statistics in this field right now. And everyone has their favorite approach that they think is right. If you're using any of these software packages, you should definitely read the paper, and you should try to dig into some of the statistics and understand it. To further complicate your life, using different approaches does indeed give you different results. It would be super nice if all these different approaches all led the same place, but they don't. So again, it's worth maybe looking around at what people are publishing in your target journal, what packages are available that somebody in your lab knows how to use and understands. But the overall story is that as you increase the number of samples, the number of detected genes in general tends to go up no matter what package you use. The other thing is that as you increase the number of samples, your precision tends to go up. So same story as last time. The more biological replicates you can get into the experiment, the more likely it is you will find real differences, and that no matter what statistical package you pick, it will be sufficient to find those differences. So I talked about this a little bit last time. Technical replicates are something you should try to control for technical variation, but I would not waste your time and money sequencing based on technical replicates, because the biological variation is so much bigger than the technical variation. At this point, it's not worth measuring the technical variation. I mean, it is if that's the question you're asking, right? If your deep burning research question is, do these two library kits differ? Or, you know, does this instrument yield a completely different result than that? If that's your question, great, measure it. But if your question is, what does my treatment do to this organism? Focus on biological variation, because it's way bigger. and that's what you're gonna have a harder time statistically controlling. So that's just my 2 cents on, and I would say there is good literature to back that up. I can point you to the citations. Yes, this is re-saying the same thing. Decisions are largely driven by limited biological replicates. Yeah, so one day when you have lots of data, non-parametric, I suspect may take over, but we are not there yet. With over $200 a library for each RNA-seq experiment, that's largely limiting how we do bioinformatics in this space right now. So a little bit more information about DE-seq2. It is important to realize that it accepts only raw counts as input. Do not put RPKMs. Do not put TMMs. Do not try to normalize your data in any way. Do not try to adjust for read depth per library. DESeq2 is this very comprehensive package, and it wants to do everything for you. So only put in raw counts. The nice thing, one of the things I like about DESeq2 is that it fits a generalized linear model, which is super flexible. You can allow for very complex design. So if your experiment has a whole bunch of different factors, you can model each and every one and the interactions of each and every one. By default, it uses the walled test. It uses an adjusted log fold change. So that's the test statistic. And the nice thing about the walled test is there's no need for a reduced model. If you want to test a reduced model against a full model, it also offers a likelihood ratio test. So you do have some statistical options there depending on what kind of questions you're trying to ask. Built in, there is adjustment for multiple testing, which is important in RNA-seq because you're generally asking all of your questions by each and every gene, which means you've got 30, 40, 50,000 individual tests, and you are going to need to adjust for the fact that you're doing all of those tests. Benjamini and Hochberg is, I think, the default standard in DESeq2, and you can adjust that to something else if you want. So here's an example. If we had, this is from the manual, if you had, let's say these are all of your RNA-seq libraries, you have 3 that were treated, 4 that were untreated. In this case, some of them are single N and some of them are paired N, which turns out to be a very important factor, probably, in your experiment you want to control for. So you can treat these as 2 different factors, and you can set up your generalized linear model with both of those factors. So you can say my model includes type and condition, but in DESeq2, by putting condition last, that becomes the variable of interest. And the results of this design, using the walled test, will return genes corresponding to the condition factor. So instead of doing something like a reduced model where you might do type plus condition compared to the reduced type model to see if condition is significant, this actually, you just put one model in, and whatever's last is what gets used as your factor of interest. You can also put into your formula an interaction term. So this would be saying, I want to look at genes that are responding to the interaction of type and condition, not just to condition or not just to type, but vary based on their interaction. And these are kind of just standard ways to specify model designs in R, but I wanted you to see them. Yeah. Because it's important often to test your data for both, right? You want to see what's responding to your factors, but also if your factors are interacting. So don't forget to do that. There are a number of great vignettes and manuals available for DESeq2. All of these things, one of the nice things about it is it gives you all these visualization tools and kind of helps you really build beautiful figures for your paper, which is great. You'll probably see MA plots in a lot of these different packages. And these are kind of fun to look at because they talk about how that variation shrinkage happens. So basically the log fold change, and this makes sense. So if your expression is very, very low, so let's say you have very, very, very few reads in a sample, your log fold change can be immense, but it's not statistically significant because you didn't sample enough if it's very lowly expressed. Like let's say you had a read count of 1 and then you had a read count of 10. That's a huge log fold change. It's 10 times more expressed, but there's so few reads. Is that really significant? So down here at this very low expression level, where you have these huge log fold changes, DESeq2 basically just shrinks all of that, all of those log fold changes, and says those aren't significant. So you can see the things that are your sweet spot for differential expression, which is everything in red. It's going to be things that are moderately to highly expressed and have moderate to high log fold changes, as you would expect. So this is just trying to visualize how much of my data ended up being so lowly expressed that I couldn't detect differential gene expression. It turns out, like quite a lot often is falls into that category. DESeq2 will help you to generate heat maps. We use something called the variant stabilizing transformation. So it turns out the way DESeq does the normalization, it doesn't make the genes directly comparable to each other. So there's an extra step to get them into, there's a number of different transformations you can pick. The manual has all the instructions. But you go through these steps and follow the manual, and you can get to heat maps. And you can actually do hierarchical clustering of the genes and the samples, so you can see which ones are most similar.
Hello.
Hi.
How are you?
Good.
My name is Nick. I'm ACF User Assistance.
Thank you.
No problem.
We will be logging on in like 5 minutes.
That's fine. Take your time.
It's gonna work today. We have an expert here. Oh yeah, like I was saying, you can get hierarchical clustering, and these make really pretty figures for your papers to see which of your samples are the most similar. And of course, you can always do PCA plots, which are great for exploring the overall variation in your data. More descriptive statistics than quantitative, but it's kind of nice because a lot of this is wrapped inside the software package, so we'll see that. I do want to talk a little bit about transcript level quantification, which we're not going to do in the lab in this class, but it's a new and very important kind of method of doing informatics on RNA-seq that's worth at least knowing about. And this is basically, if you work on a really well described organism, your genes may have multiple isoforms that are known and described, so multiple alternative splicing possibilities. And we talked about this a little bit in the last class, where you can use your paired-end reads to identify, maybe potentially uniquely identify these isoforms from the reads. Sometimes you have reads, like the purple reads, where they fall into regions that are shared on both isoforms, so they are not helpful. You do not know which isoform. But for the red and the blue reads, you can, based on the distance the reads are apart, based on where they map, you can actually determine if you have isoform A or isoform B of a gene. And this is kind of an interesting question, because there is different isoforms splicing based on different, that can be very important for gene regulation and gene expression, and how your organism is responding to whatever the treatment is. Oh, and length, right? I talked a little bit about how like gene length does matter. Sampling gene length is something you might want to normalize for. If this isoform is clearly much shorter than the other isoform, then normalizing on the length of that isoform would be important, because you are potentially trying to compare these to each other because they're from the same locus. Yeah.
Why do you normalize the read length selected while preparing the library to give like size selection? Why do you need to normalize read length?
So you're not normalizing based on your read length. If I said that, I misspoke. You're normalizing based on the whole transcript length, the original biological transcript, right? Because once it got fragmented and then you size selected it, if it's thousands of bases, you're gonna have more fragments than if it's a short little gene, you only have a couple fragments. So that would change how much of it you would sample in your sequencing experiment. So yes, you are doing that. You are pulling down fragments of the size you want for sequencing, but there may be more of them if it's from a bigger original transcript. Yeah. So there are plenty of software packages to calculate transcript abundances. So this is based on the transcript, not the gene, right? You can multiple transcripts per gene. The biggest ones are salmon, sailfish, Callisto, and RSIM. We've used salmon a little bit in my lab. It's super fast, it's really nice. You can still use DESeq2. However, DESeq2 expects gene level counts, not transcript level counts. And there's still, I think you can actually force it to do transcript level statistics, but that's not what it's meant for. Something like RSIM is probably what you want if you want real transcript level abundances. There's this really interesting paper basically saying that gene level results are often more accurate, powerful, and interpretable than transcript level results, because the transcript level results get messy, especially if you don't actually know all the isoforms of your gene in the organism. But if you incorporate transcript level estimates, it actually yields more accurate gene level results. And so if this is something you're interested in, if you work in like Drosophila or mouse or something that you would actually have this type of data, I will send you this paper because it's really interesting. If you're working in something where you don't have any of that, then don't worry about it. If you have a de novo assembled transcriptome, this does present some statistical problems. You often have lots of little pieces of the same gene floating around in your transcriptome, which is basically means fewer reads can be aligned unambiguously, and your statistical power has gone way down. So I would say put some money into trying to get the best de novo transcriptome you can. There's a new PacBio protocol called IsoSeq that can really help you develop a rock-solid transcriptome reference that is would help to combat this. If you can't do it, basically you're going to need more data to get the same amount of power as if you had like a real set of gene models. So just a warning there. And if you want to talk about numbers, like what kind of reads does that look like, let me know. One thing that is really helpful, and I do advocate if you have a de novo transcriptome, is to use either Corset or RapClust. We really like RapClust in my lab. And it has this, I do like the name Corset though. I feel like Corset is a much better name than RapClust, especially because they put this diagram in their paper which looks like a corset. But basically, you take your de novo assembled transcriptome, you map all your reads, and then it uses that paired in read mapping information to try to figure out which transcripts originated from the same gene. So instead of doing your statistics on each individual transcript, it creates a whole group of transcripts that it calls a gene, and it aggregates all those read counts to make your statistics more powerful. And then at that point, you can perform your statistical testing on these like transcript network units instead of on each individual transcript. And it helps to make your de novo transcriptome assembly behave much better. So those are really good. Here are lots of references for people who are doing this and want to do some more reading. Do you guys have any questions? That's an extremely high level overview of the statistics, which are actually surprisingly complicated. But you know, at least you have a little bit of knowledge about it now. Any question on approaches, thoughts? All right, we will go ahead and switch over to the lab now. Did you want to use my computer? You do? You're going to use yours? Okay.